All right, today we're jumping into a real-life viral detective story that actually unfolded on poultry farms. In this case, it's got everything: a mysterious killer, a super complex genetic puzzle, and of course, a race against time to figure out a new and evolving threat. So it all kicks off with this one really troubling question. You have these farmers who are doing everything by the book, right? They're vaccinating their flocks just like they're supposed to, but the chickens, they're still dying. So what on earth was going on? So our story really begins on the ground, right in the middle of a pretty serious outbreak in Egypt that had veterinarians and farmers just completely stumped. Yeah, so across 30 different farms, this deadly pattern starts to emerge. We're talking up to 35% of the chickens dying. They're showing these awful symptoms like dehydration and diarrhea. But here's the part that makes it a real puzzle. Every single one of these flocks had already been vaccinated against a known killer virus, IBDV. I mean, that vaccine was supposed to be their shield, but it was just failing. That's a terrifying thought for any farmer and a massive red flag for scientists. Okay, so what exactly is this main suspect IBDV? Well, think of it as a virus that goes straight for the chicken's immune system headquarters. It's incredibly contagious, it can be absolutely devastating for young birds, and it's one of the most economically damaging diseases in the whole poultry industry. It's a big deal. So, with the vaccines clearly not working, scientists knew they had to dig a lot deeper. It was time to try and crack the virus's genetic code. The investigators went out and collected tissue samples from the sick and deceased birds. The plan was basically to perform what you could call genetic fingerprinting on the virus itself. They were hoping to find some kind of clue hidden in its DNA, or to be more precise, its RNA. You see, the IBDV genome is split into two really important parts. You can think of segment A as the virus's make and model. It tells you what type of virus you're looking at. Segment B, on the other hand, that's more like the engine. It's directly connected to how deadly, how virulent the virus is. To solve this mystery, the scientists had to look at both. And when they got those genetic fingerprints back and analyzed them, they found something totally unexpected. This was not the usual suspect they were looking for. So check this out. This is the genetic family tree for segment A, the make and model part of the virus. And it gave them their first huge clue. You see, 5 of the 6 samples grouped exactly where they expected them to, over on the right with the known very virulent killer strains. But look closely at that one sample right there, MANS 1, marked with a blue triangle. It's a total outlier. It's hanging out over on the left with much older, classic versions of the virus. But here comes the twist. When they looked at the family tree for segment B, the engine part, the story completely changed. All 6 samples, including our weird outlier MANS 1, were grouped together smack in the middle of the very virulent category. So what that meant was that this strange virus, MANS 1, had the engine of a stone-cold killer. And this table just lays it out perfectly. The usual killer virus, the one they expected to find, has a very virulent type and a very virulent engine. Simple enough. But this new hybrid? It combined the classic virus type with that very virulent engine. They had just discovered a brand new kind of viral hybrid. So what does this actually mean? Well, viruses can do this thing called reassortment, where they swap genetic segments. It's basically a genetic mix and match. In this case, it was like the virus took the body of a classic car, but then dropped in the engine of a high-performance race car. This kind of evolution is exactly how new threats pop up that can catch our current defenses, like vaccines, totally off guard. Discovering a new hybrid virus like this immediately raises one giant critical question. Okay, so just how dangerous is this new thing, really? To find out, the scientists set up a head-to-head competition. They got 3 groups of healthy, susceptible chicks. Group 1, they were the control group, left untouched. Group 2 got infected with the new hybrid virus. And Group 3, they were infected with the known superkiller strain. Then for 7 days, they just watched to see what would happen. And the physical evidence was pretty grim. This here compares the internal damage caused by the 2 viruses. So on the left, you can see some pretty significant muscle hemorrhaging from the new hybrid virus. Now look at the right. That's the damage from the known killer strain. And while both are obviously causing serious harm, that tissue damage on the right just looks more widespread, more severe. So, after those 7 days of observation, the experiment was over, and it delivered a very clear verdict on the threat level of this new hybrid. And the hard numbers, the quantitative data, it confirms what those pictures were suggesting. This chart shows the average tissue damage scores, and you can see that for every single key organ, the chickens infected with the known very virulent strain, that's the bar on the right, showed way more severe damage than the ones that got the new hybrid. But what about the ultimate measure, right? Mortality. Well, the new hybrid virus was incredibly lethal. It killed 60% of the chickens it infected. That is a truly devastating number. But the original, very virulent strain, it proved to be even deadlier with a mortality rate of 70%. So the verdict is in. This new hybrid virus is absolutely a serious, deadly threat. No question about it. But the study concluded that the established, very virulent strain is still public enemy number one. It causes more severe disease and, in the end, slightly higher mortality. So why does this one case on a bunch of chicken farms in Egypt matter to the rest of us? Well, because it's a perfect illustration of a universal truth. Viruses are always changing. This genetic mix and match thing they do creates new threats that can just bypass our defenses, like our current vaccines. And that's why continuous surveillance isn't just some academic exercise. It's absolutely essential for protecting animal health, which, is directly tied to the stability of our entire food supply. And that really leaves us with this final, crucial question. In this constant arms race against an enemy that is always evolving, where new hybrids can just pop up at any moment, how in the world do we stay one step ahead?